Okay, let's try and unpack this one. Imagine an enzyme you think you know, right? Like one famous for copying RNA into DNA, like you see with HIV.
Hmm.
Now, picture it doing something, well, utterly bizarre. Instead of needing a template sequence, it just starts building these massive chains of DNA.
Right.
But they're made of only one building block, just one letter, repeated over and over, like hitting repeat, seemingly out of thin air.
Yeah, it's pretty wild.
And that's just one piece of the counterintuitive molecular arms race we're diving into today. Welcome to the deep dive.
We're exploring how life, from the tiniest bacteria right up to our own cells, gets incredibly inventive, finds these totally unexpected ways to fight off invaders.
And our sources today are two really fantastic recent papers, top journals, Nature and Cell.
Absolutely top-tier stuff.
And our mission, as always, is to take these complex findings, try to strip away some of the jargon, and really reveal the key insights, those aha moments.
Yeah, and show you why they matter. Whether you're, deep in the biology trenches yourself, or maybe just intensely curious about these hidden battles happening constantly inside living things.
So maybe set the stage just a little.
Sure. So just briefly, remember that living things are, they're in a perpetual battle against threats. Bacteria, for instance, constantly targeted by viruses. We call them phages.
Right, phages.
It's this ancient conflict, right?
Right.
And bacteria have evolved all sorts of defenses. Physical barriers, sure, but also sophisticated molecular tools, like CRISPR systems that snip up viral DNA. You've probably heard of CRISPR.
Yeah, CRISPR's pretty famous now. And our own cells have innate immune systems too, don't they?
Exactly. We have sensors like cGAS, usually patrolling the cytoplasm, the main body of the cell, ready to sound the alarm if they spot foreign DNA just, hanging around where it shouldn't be.
Okay.
And then there are reverse transcriptases, RTs. Normally we think of these enzymes, copying an RNA sequence to make a matching DNA sequence, using that RNA strand as a very precise template.
Like retroviruses do to get into our genome.
That's the classic example, yeah. They use it to integrate their genetic material.
But what if an RT wasn't just copying? What if it was, synthesizing DNA in a way we just hadn't imagined?
Exactly. And that's where our first deep dive takes us, right into that Nature paper.
Let's get into it.
Right. So the researchers, they were looking into this family of bacterial defense systems called DRT. Stands for Defense-Associated Reverse Transcriptase.
Okay, DRT.
They knew one type, DRT2. It worked pretty much as expected. Made antiviral DNA using an RNA template, like a typical RT.
Standard stuff.
But the most common type, DRT9, that was a complete mystery. It didn't have the usual partner gene nearby, the effector gene that codes for the protein that actually does the fighting.
So the RT was there, but the weapon was missing.
Kind of, yeah. So they asked, how does DRT9 actually defend against a virus? Does it even make DNA? And if it does, what kind is it, and how does it work?
And this leads to that really astonishing result we mentioned at the start.
It does. They infected the bacteria with phage, and boom, the cells showed this massive, rapid burst of really long DNA chains, like over 10,000 base pairs long. Huge.
Wow.
And when they analyzed these chains, this is the kicker, they found something totally unexpected for DNA. They're made up only of the DNA base A adenosine, pure poly-DA.
Just A's, like A for thousands of letters.
Exactly, just A's. And they confirmed it was DRT9 doing it because when they knocked out the DRT9 gene, genetically removed it, this whole poly-DA production just stopped, dead in its tracks.
So that's the polynucleotide bomb.
That's what they called it, yeah. It's like the bacteria suddenly starts churning out these long strings of just one type of chemical building block.
Okay. So they're making these, bizarre poly-DA chains. But the massive question is how? How does a reverse transcriptase, which is supposed to need a template, make DNA without one? And why only A's?
Right. And the paper just beautifully unpacks this mechanism. It's like peeling back the layers of an onion.
So how does it even start the chain?
This is maybe the most mind-bending part. Usually, DNA synthesis starts by adding bases onto an existing primer.
Right.
A little piece of DNA or RNA.
Yeah.
But here, the enzyme itself, the DRT9 protein, kicks things off. A specific amino acid, a tyrosine, right at the very beginning of the protein chain, the N-terminus, it acts as a protein primer.
Wait, the protein is the primer?
Yeah. It forms a direct chemical bond, an O-ether linkage they found, with the very first adenosine nucleotide, DAMP.
Whoa, that just throws the usual rules for starting DNA synthesis right out the window.
Pretty much. And they proved it really elegantly. They mutated that one tyrosine, changed it to a different amino acid, phenylalanine, which is similar but lacks that key oxygen group.
Yeah.
And the whole synthesis process just shut down. completely.
Yeah, okay, so clear proof that tyrosine is absolutely essential.
Definitely. So the protein primes it.
Yeah.
But what about the RNA? There's an RNA involved too, right?
Yeah, wasn't it supposed to be the template?
Well, that's the twist. It's crucial, but not as a sequence template. They use cryo-EM, which is like taking these incredibly detailed 3D snapshots of molecular machines.
Amazing tech.
It really is. And they saw that DRT9 protein forms a hexamer, a complex of 6 identical protein units arranging themselves together.
Sticks of them. Oh,
And a specific non-coding RNA molecule, it wraps around this hexamer complex.
so the RNA is more like a scaffold, a
Sure.
structural guide.
Precisely. It acts as a scaffold. It holds those 6 RT units in a very specific circular arrangement. And inside this complex, the RNA even folds into a triple helix structure, which is pretty unusual in itself.
Go helix. Okay.
And it seems this specific structural setup, the way the 6 RTs are positioned and interact within that circular channel defined by the RNA scaffold, that's what guides the addition of only DA nucleotides, clockwise around the channel.
So the structure forces the enzyme to only pick A's.
That's the model. The structure dictates the substrate choice and the direction. That's how you get a pure poly-DA chain without a matching template sequence telling it what to add.
That is incredible molecular engineering by these bacteria. Just wow. Okay, so they build this massive poly-DA chain. What does it actually do? How does that stop the virus?
It's what's called a sacrifice protection strategy. Pretty common in bacteria, actually.
Okay.
The rapid massive synthesis of these huge poly-DA chains, it acts like a chemical vacuum cleaner. It very quickly sucks up the cell's entire supply of DATP.
DATP, that's the adenosine triphosphate nucleotide, one of the 4 DNA building blocks.
Exactly, the A building block. Phages desperately need lots of DATP to replicate their own DNA.
Right, so you starve the virus of its essential supplies.
That's the idea. By depleting the cell's pool of DATP, the DRT9 system effectively slams the brakes on the phage's ability to copy its DNA and make new viruses.
Okay.
And this stalled infection, it then seems to trigger the infected bacterial cell to undergo programmed cell death, basically suicide.
So the infected cell dies.
It dies, yes. But by doing so, it stops the phage from multiplying and bursting out to infect all its neighbors. It sacrifices itself to protect the whole bacterial population.
A real single-cell hero act for the good of the community. Amazing. And it's not just sitting there waiting, is it? There's some regulation.
Oh, yeah. There's this fascinating molecular tug-of-war going on.
Okay.
The viruses, they've actually evolved a way to trigger this defense. Maybe as a last resort, or maybe it messes something else up for the bacteria. We don't know for sure. But they have a small peptide, a little protein fragment, that can bind to the outside of that DRT9 hexamer complex and switch on the poly-DA synthesis.
The virus activates the bacteria's self-destruct.
It seems so. But the bacteria have fought back. They've counter-evolved a host protein called SID. And SID competes with that viral peptide. It tries to bind to the same spot on the hexamer.
Ah, competition.
Exactly. SID acts as an inhibitor. It keeps the system off, unless the viral trigger peptide is there in high enough amounts to win the binding competition. It's this really elegant molecular battle deciding who controls the switch.
Wow. That is intricate. Figuring out a mechanism that complex protein priming, RNA scaffolding, the hexamer structure, the nucleotide selection, the regulation, that must have taken a huge team effort.
Absolutely. This Nature paper is just a perfect example of multi-institutional collaboration.
Yep.
You had teams pooling expertise from Columbia, Montana State, Berlin, Brown, all over.
Yeah.
They used everything. Genomics to find the system, biochemistry for enzyme function, cryo-EM for the structure, mass spec to nail down that protein primer, even computational modeling to put it all together.
And you really have to admire the scientists involved. I mean, Samuel Sternberg's lab, they have a track record for finding these weird roles for RTs and immunity, right? So he clearly had the intuition.
Definitely. And Stephen Tang using single molecule fluorescence, actually watching the synthesis happen in real time, that's pushing boundaries, bridging chemistry and physics.
But what really jumps out, and this is just so crucial in science, it's that they noticed that abnormal fragment, that pure DA chain. Like you said, maybe other researchers would have just dismissed it. Oh, that's contamination or artifact.
Right. It's easy to ignore weird results.
But they saw something strange and had the persistence, the curiosity to dig deep. And that's what led to this amazing discovery.
Absolutely. That ability to see the unexpected and chase it down is key.
Okay. Let's switch gears completely now. Our second deep dive, this paper in Cell, this one takes us inside our own cells, into the nucleus, actually.
Right.
And it looks at how viruses might be triggering our innate immune system from a totally unexpected location.
Yeah, this one started with a real puzzle about cGAS,
Yeah.
that immune sensor we mentioned earlier.
The one that detects DNA, usually in the cytoplasm.
Exactly. But researchers had noticed something odd. Some viral proteins seem to activate cGAS inside the cell nucleus.
But wait, the nucleus is packed full of our own DNA, right? Wound up tightly in chromatin. How do viruses expose DNA there to trigger cGAS without our own DNA constantly setting off alarms? That seems like a big problem.
It is a big problem. And the key discovery in this paper is that certain viral proteins, they looked at examples from common viruses like herpes simplex virus 1, HSV1, and cytomegalovirus, CMV.
Okay, common ones.
They don't just randomly expose DNA. They specifically target a protein found at a very particular, very important place on our chromosomes, the centromeres.
Centromeres, those constricted bits in the middle of chromosomes.
Yes, exactly. They found these viral proteins interact directly with a protein called CENP-B. It's a key component of centromeres, absolutely vital for making sure chromosomes get sorted correctly when cells divide. The viruses are targeting this really critical piece of cellular machinery.
And messing with CEMP-B at the centromere, that causes chaos.
It seems to. It triggers what the authors call a central burst.
Centroburst, okay.
What happens is specific DNA sequences right there at the centromeres, these highly repetitive sequences called alpha satellite repeats, they get amplified dramatically. Many, many more copies are made than normal.
Okay, lots of extra centromere DNA.
And the model they propose is that this excess alpha satellite DNA then gets sort of pushed out from its usual tight packaging. You know, the nucleosomes where DNA is wrapped around histone proteins.
So it becomes unpackaged, naked DNA.
Exactly. It forms these loops of exposed naked DNA right there within the nucleus, but specifically originating from the centromere.
So the virus causes a localized explosion, like an unpacking of DNA, right at these specific chromosomal spots.
That's the idea. And this exposed centromeric alpha satellite DNA, that's what then attracts cGAS into the nucleus and flips its switch, activates it. Activated cGAS then does its job, synthesizes that signaling molecule, cGAMP, which basically broadcasts the danger signal to the rest of the cell's immune system.
And they had proof for this, that it was really CENP-B and the centromere.
Yeah, they provided strong evidence. They showed that if you get rid of CENP-B, or another protein called TRF2 that's also found around centromeres and telomeres.
Telomeres, the chromosome ends.
Right. If you remove those, this whole process, the DNA amplification, the cGAS activation, gets blocked.
Wow. Okay. And this isn't just about fighting viruses, is it? Does this connect to human diseases, too?
It does, potentially. The paper showed something really interesting using human-induced pluripotent stem cells. They used cells lacking an enzyme called TREX1.
Okay, TREX1.
Yeah, losing TREX1 is linked to some severe autoinflammatory conditions like Aicardi-Goutieres syndrome. It's kind of a genetic model for those diseases.
Right.
And in these TREX1 deficient cells, if they artificially force the amplification of those same alpha satellite DNA sequences.
The ones from the centromeres.
Yes. It triggered a strong immune response, and it also messed up how the cells differentiate, how they specialize. And those effects mimic some key aspects of those syndromes.
So problems at the centromere may be triggered by viruses or maybe other things could potentially be involved in some of these inflammatory or developmental disorders.
It certainly suggests a possible link, yeah.
Yeah.
That uncontrolled centromere DNA exposure could be a contributing factor. It's a really intriguing lead.
Another great example, like the first paper, of different fields coming together. Virology, chromatin biology, innate immunity.
Absolutely. A really powerful alliance of research groups. From places like Rockefeller, Pasteur, Heidelberg, you need that combined expertise.
And again, you have to appreciate the scientists. Someone like Francois Lahaye, apparently he's been working on nuclear innate immunity for a decade.
Yeah, it takes real insight and, frankly, persistence to even hypothesize that viruses might target something as fundamental, as conserved as the centromere to trigger immunity. That's not an obvious connection.
Yeah, not at all.
And then to have the technical chops using systems biology and other approaches to actually validate that hypothesis. That's great science.
Okay, so let's try and bring these two deep dives together now. What's the big picture here? How do these studies really change our understanding, and maybe what do they inspire for the future? Let's start with the innovation. How innovative was that DRT9 study in Nature?
Oh, it's fundamentally innovative in several ways, I think. First, it brought protein-primed DNA synthesis. that idea of the protein itself, starting the chain into the world of bacterial reverse transcriptases.
Right, that was mostly known from viruses before.
Exactly. So it hugely broadens our view of what RTs are biochemically capable of. They can do way more than just straightforward template copying.
And it also expands this idea of synthetic immunity, right? Where the cell actively builds molecules to fight invaders.
Yeah, but instead of building something complex, it's building these, well, strange homolomers, just strings of one base. That's a new twist on synthetic immunity.
Okay, what else?
Well, the mechanism itself was completely novel. The combination of the RNA scaffold structure, the tyrosine priming, and the way the 6 units in the hexamer work together synergistically.
Getting that first structure was key.
Absolutely. Seeing that 3D structure of the DRT9 system let them put all those biochemical pieces together into a coherent mechanism, that was a major breakthrough.
And from the bacterial perspective, how does it change how we think about their defense strategies?
It adds a whole new weapon to their arsenal. Bacteria aren't just, chopping up viral DNA with restriction enzymes or CRISPR or blocking the virus from getting in.
Yeah.
They can use this strategy of nucleotide exhaustion, basically starving the phage by sucking up all the DATP, and couple that with programmed cell suicide. It's another powerful anti-phage tool, especially thinking about protecting the whole population. It definitely enriches our understanding of their defense playbook.
Okay, so looking at both papers now, what kind of future research, what new directions do they point us towards?
Well, from the DRT9 study, that protein-primed RT itself is just fascinating from a biotech angle. Think about it. An enzyme that builds single-base chains, no sequence template needed, carries its own structural RNA guide. Seems to have built-in regulation.
Yeah, it could be engineered.
Maybe. Could you use it for stuff like adding specific tags to molecules in the lab? Or, I don't know, maybe even building simple nanomaterials out of pure base chains. It's kind of out there, but the properties are interesting.
And what about the biological questions it opens up? Still lots to figure out there, I bet.
Oh, so many. Like, how do the uninfected bacteria in the population tolerate this poly-DA synthesis if that SID inhibitor fails? How common are these DRT9 systems really? Are they in lots of different bacteria, different environments?
And how do they play with other immune systems?
Exactly. How do they interact or coordinate with other defenses like CRISPR, which you often find coded nearby in the genome? Is there crosstalk, cooperation, interference? Lots to explore.
And turning to the cell paper, the centromere discovery, what future work does that inspire?
Well, a huge area is obviously exploring if this centro-burst mechanism is involved in other autoimmune or autoinflammatory diseases, or even neurodevelopmental ones, especially any that have a suspected viral trigger or known issues with DNA metabolism.
Beyond the specific syndrome they modeled.
Right. Can we find more viral proteins that target CENP-B or maybe other centromere proteins? How do these viral strategies evolve over time? And crucially, could we find ways to disrupt them therapeutically?
It feels like ultimately both of these studies, they really underscore something pretty profound about how life works.
I think so. They just showcase the incredible diversity, the ingenuity, the sheer complexity of the molecular strategies happening inside living things, often in places, like the nucleus or using mechanisms we never even thought to look for.
Yeah, bacteria building DNA bombs in this totally unexpected way.
Or viruses hijacking these absolutely fundamental parts of our own chromosomes, the centromeres, to trigger an immune alarm.
It's just a powerful reminder that even in areas of biology we think we understand pretty well.
There's still so much left to discover. And these fundamental biological processes, they could be repurposed and reused in the most surprising, almost inventive ways.
Okay, so to wrap up this deep dive then, we've explored bacteria using this previously unimagined template-free DNA synthesis coupled with cell suicide to fight off phages.
And on the other side, viruses potentially triggering nuclear immunity inside our own cells by specifically targeting and exposing DNA right at the centromeres.
Two really cutting-edge examples showing just how dynamic, how inventive biological systems are in this constant arms race against invaders.
Constantly evolving, constantly surprising us.
So here's something to think about. If a bacterial enzyme can build these massive DNA chains made of just one letter with no sequence template, using the protein itself as a starter, what other seemingly impossible molecular feats are happening inside cells right now that we haven't even begun to imagine?
Yeah, or maybe, could understanding exactly how viruses can trigger these problems at our centromeres, could that one day give us vital clues to understand and maybe even treat really complex conditions like certain inflammatory or developmental disorders.
Lots to ponder there. Thank you for joining us for this deep dive into the surprising frontiers of molecular warfare.